Hello, my name is Jeff Kordower. I'm the founding director of the ASU Banner Neurodegenerative Disease Center at Arizona State University, and it's my pleasure to speak to you today on animal models of multiple system atrophy, genetic, proteopathic, and viral vector-mediated rat and non-human primate models. It's essential that we create valid and reliable animal models to study multiple system atrophy. I'm sure everyone in this audience realizes the natural history of multiple system atrophy with autonomic dysfunction, Parkinsonism, and cerebellar features, highlighting the two different major foci of MSA, MSA-C for cerebellar damage, and MSA-P for Parkinsonian features. All I'm going to be speaking about today is MSA-P. I'm going to talk about the different features of the disease and how it's modeled in rodents and non-human primates. And it's essential that we create good models of this disease, because unlike Parkinson's disease, where people, for the most part, live a normal lifespan, MSA is a horrible disease in which between 6 and 9 years post-diagnosis is typically when sudden death occurs. So we all know that MSA's pathology is glial cytoplasmic inclusions that are stained for alpha-synuclein. You can see a control brain on the left and an MSA brain on the right. And you can see on the right, you can see the oligodendroglial cells that are stained with phosphorylated alpha-synuclein. That's the pathological form of alpha-synuclein. And for the two different types of MSA, the MSA-P on top, where you can see the spreading in the basal ganglia, and less so in the cerebellum. But in MSA-P, it starts, as we progress clinically, it starts in the cerebellum and ends up spreading to a variety of regions, including the cerebral cortex. So we believe that MSA is an oligodendrogliopathy with secondary neuronal degeneration. So the initial insult, we think, occurs in the oligodendrocyte. There is some controversy as to whether alpha-synuclein is made in the oligodendrocyte or whether it's transferred from another cell compartment. And we'll talk a little bit more about that as we go forward. And so this is really the pathology that we're addressing. There are clinical trials ongoing which are based at trying to remove this oligodendrogliopathy. And we'll speak a little bit about that towards the end of the presentation. For many years, toxins were used to create animal models of MSA. These are based upon where cell loss is seen. So there would be 6-hydroxydopamine lesions along with quinolinic acid lesions of the striatum, but none of those recreate the alpha-synucleinopathy that one sees in multiple system atrophy. And so these are no longer in favor and are rarely ever used. So what are the models that are being used? Initially, transgenic models of multiple system atrophy are being used and continue to be used. They are 3 different promoters that in transgenic mice. The PLP promoter, which you can see here, has glial cell GCI formation, about 20% loss in the substantia nigra, microglial activation, mild motor impairment, and no demyelination. Now, demyelination is an important feature of MSA, and so this particular promoter does not recreate that aspect of the disease. There's myelin basic protein promoter, where you get a little bit more GSI. You get GSI formation, about 40% loss in the striatum and nigra, decreases in GDNF, myelinopathy, and variable motor impairments. There's a loss of oligos, which you don't see in MSA, and also olfactory deficits. And then there's the CMP promoter. And this really is not a very useful aspect for MSA, because the primary pathology is in the spinal cord, and that's where the motor impairment occurs, and that doesn't look very much like what is seen in human MSA. Up here in the top left, you can see a recapitulation of human alpha-synuclein expression in these animal models. You have the MSA brain on top, and you can see these different, with different stains looking at these GCIs, and that is recreated in the myelin basic protein transgenic animal that's seen in the lower level here. What is unlike human MSA is that these GCIs are found everywhere, and there's more regional expression in human MSA, but this is not something that's been really addressed that much in the animal models. And as we improve these models, maybe more regional expression will be, and regional expression for cell loss will be obtained. In terms of function, these animal models show motor impairment on the POL test. You can see here at 3 months, not much change, but 6 to 12 months using this particular model, we see myelin basic protein transgenics improve, have more time on the POL test than on the in comparison to the non-transgenics. So these progressive motor behavioral deficits are a good start for using transgenic models for MSA. Now, I'm not going to go into all the animal models, but I did want to discuss a little bit the work of Stan Prusner, who is used MSA extracts both from human MSA cases and also mice that are transgenic for alpha-synuclein, and put them into transgenic mice. And you can see here this, just the MSA1 and MSA10. This is control case. This is what a control human case looks like. This is what the MSA case looks like. And you can see the loss of nigral neurons in the substantia nigra, and you can see the inclusions that are found here in the different MSA donor tissue, if you will. And you can see here on Western blots the amount of alpha-synuclein that one can generate using these tissues. You can see FC is frontal cortex, SN is substantia nigra, and you can see frontal cortex in a control here, and then in the basal ganglia, substantia nigra of MSA patients, it's lower from the MSA patients here. And then you can see other MSA patients. So there's quite variability in the amount of alpha-synuclein that's seen in specific regions of MSA patients. Now, one of the interesting things is that when put into the parietal cortex of rats, you can see that if they're transgenic homozygotes, there's a tremendous decrease in the amount of the lifespan of these animals, and controls continue to go on for over 2 years. But if you give these materials into these animals, there's a decrease in lifespan. If you look at the reticular formation, you can see lots of alpha-synuclein, but the problem here is that it's all neuronal. The propagation of these extracts is not from oligo to oligo, but it's from these materials into neurons, and that doesn't really mimic what we see in MSA. There is a lot of MSA around, a lot of synucleinopathy around, and you see it in the cortex of thalamus, subthalamic nucleus, periaqueductal gray reticular formation, brainstem, and you see very similar patterns, whether they were inoculated with transgenic tissue from transgenic MA3 mice or whether human MSA extracts. Now, one of the interesting things about these particular studies is that the level of infection using the MSA extracts is much greater than using Parkinson's disease extracts. And you can see here just on panel B, you can see that the MSA mean cell infection in an in vitro study is much higher than what you see in Parkinson's disease, even though Parkinson's is regarded to be a prion-like spreading event. So that's an interesting point here using this model, but still the aspect of it transmitting from the site of inoculation to neurons, and it doesn't really make this a viable MSA model going forward. And just to reemphasize this point, if you look at these Western blots on the left and the quantification on the right, and if you look at all these regions here with MSA, the amount of phosphorylated alpha-synuclein goes up often more than tenfold. But if you do the same kind of studies in Parkinson's disease, you see that they are very low levels of phosphosynuclein here. And again, just to show you that in controls, there's very little spreading of phosphosynuclein, but and very little spreading in Parkinson's disease case 1 or Parkinson's disease 2. You look at the astrogliosis, there's virtually none. But if you look at the MSA cases, there's much more phosphosynuclein, and there's much more astrogliosis, and there's much more microgliosis as well. So these are the rodent models that have been existing and have been being developed over the last 10 years or so, especially the proteopathic models that Sam Prusner has led. I'm going to now talk mostly the rest of the way about work that we're doing. And also this has been done by Erwin Bizard and Vasilis Meisner from Bordeaux in their studies using a slightly different vector. But our goals is to deliver alpha-synuclein using a specific vector that only transfects oligodendroglial cells. And we're doing 2 sets of experiments. One set of experiment is a viral vector-mediated overexpression of alpha-synuclein in striatal oligos. And we're going to look at time-dependent axonal and neuronal degeneration. I will tell you right now, we've looked at motor deficits, both in rodents and in our second name non-human primates. Our lesions are just not big enough yet. We're on the precipice of getting motor deficits using this model, and we think we'll be successful in the future. But we have not done this. We have not seen motor deficits in this model yet, but I will show you from an anatomical point of view. We believe that this viral vector model is spectacular, and we'll walk you through that. So Steve Gray and Tom McCown were giving a presentation at a gene therapy meeting, and my colleague Ram Mandel just randomly walked into this session, and what they found was that they created this model, they created this vector that only infected oligodendroglial cells. Now, Steve Gray was giving the talk. He didn't know very much about MSA. My colleague walked up to him and says, you know, that's how MSA works. Would you want to collaborate? And Ron called me. And so we have a vector called OLIG001. It's a novel oligotropic adeno-associated virus. It was engineered by capsid shuffling and direct evolution. So basically just throwing all these capsids together and testing them and see whether it gets what you want. So it's a chimera capsid of AAVs 1, 2, 6, 8, and 9, and it's driven by the chicken beta actin promoter. So when we first put this in rodents, we were amazed at what we saw. First of all, we did it in 2 ways. We did it on the left with GFP. This is actually all GFP. We did it with fluorescence, and we did it with diaminobenzidine, looking at GFP. And you can see here, look at the internal capsule fibers and fascicles that course through the striatum of a rodent. And you can see how lit up they are because all the oligos in this region are transfected with this vector, as is the overlying corpus callosum. And you can see this here as well. Actually, this on the right here is alpha-synuclein, this GFP. In the GFP, you can see we have 150,000 cells surviving for the most part in these 4 rats here. And we have a significant volume of the striatum that is taken up by this vector. In contrast, well, not in contrast, but with the alpha-synuclein. If we look at serine 129 phosphosynuclein, you could see over 800,000 surviving cells that are transfected in rat 1, about 600,000 in rats 2 and 3, and then smaller numbers in rats 4 and 5. But still very large numbers of cells that are transfected and very large numbers of cells expressing alpha-synuclein in the oligos 4 weeks post-injection. So then we wanted to confirm the tropism that we saw on the slide previously. And you can see where we double labeled for GFP and OLIG2, which is an oligodendroglial marker. And you can see GFP and OLIG2, when it's merged, almost all the cells are transfected. You can see the yellow here. If you look at GFP, excuse me, you look at GFP and nu N, looking at transfection in neurons. Very rarely do you see any positive transfection. There's one cell here. And if you look for transfection for GFAP, again, very little, very few cells. And here's the quantification on the graph down below. And it's 94 to 97% oligos, about 2 to 5% neurons, and less than 1% astrocytes. So things that we wanted to do, this vector is doing, and now we're ready to go into some of our other more relevant studies. Okay, so titer-dependent expression and degeneration. So at 5 months post-injection, we put in 2 different titers of this vector. 2.4 times 10 to the 11th is our low titer, 2.3 times 10 to the 12th is our higher titer. And you can see here first with GFP, you can see very good expression. And then low titer oligodendroglial cells. We have some alpha-synuclein, serine 129 alpha-synuclein inclusions, but the high titer does better. And you can see a lot more transfection here in these oligodendroglial cells. And after 5 months post injection, you can see there's a small but statistically significant loss of nu N positive cells in the striatum, much like you see with in human MSA. This, in fact, this decrease is actually much bigger, because what we did not do is just outline the area of transfection and count the numbers of nu N positive cells there. We counted everything in the striatum, and you can see there are regions of the striatum that are not transfected. So the level of nu N cell loss here is actually much larger than what one sees. One of the interesting things is we're able to see spread of the transfection around the basal ganglia. Here's LB509, which is for total alpha-synuclein. You can see this at the injection site. And then the targets of the striatal neurons, the globus pallidus, seen here in panel B, and also the substantia nigra, which receives striatonigral innervation, is also very heavily transfected from just a striatal injection. And when you inject these cells with alpha-synuclein, and you can see here they're stained for LB509, and they double label with OLIG2, authentic GCIs are seen at least 5 months following the injection in rats. Also, other transcription factors like P25A, which is found in human MSA, is stained following these injections, and it co-localizes with the LB509. So P25A, which is a pathological feature of human MSA, is also seen within this animal model. And then you can just see here staining for LB509, especially in the areas of the corpus callosum and white matter bundles. You can see the robust number and neuropil of oligodendroglial cells that are now transformed to have alpha-synuclein. And some of these are proteinase K resistant, but some are not. And that tells you that these are authentic GCIs that are true aggregates of oligodendroglial cells and alpha-synuclein. One of the important features of multiple system atrophy is demyelination. And you can see here when we inject into the corpus callosum, or just above the corpus callosum here, you can see Luxol fast blue, a marker for degeneration, is seen right in the area of the P1 phosphoserine 129. But if you do the same thing with GFP, you don't see any, and here's where the GFP is, you don't see any loss of Luxol fast blue. This is in the corpus callosum. In the striatum, you see very similar things, and you can see the loss of myelin here with Luxol fast blue in rats that are injected with oligo-alpha-synuclein, and this is completely not seen in rats that received a oligo-1 GFP. And you can see on electron microscopy, this in collaboration with Dan Nicholson, that there's an unraveling of myelin in this model, very similar to the unraveling of myelin that one sees in human multiple system atrophy. Also, there's spreading from oligos to nigroneurons, and Patrick Brundon's lab has shown this in other systems. And what you can see here is that in the Sphancia nigra, there are cells that are olig-1 and serine 129 labeled, and they double label with TH positive cells here. So there is some spreading from oligos to neurons. It's not a lot. It probably doesn't impact much of the pathology that we see here, but it is something that does occur. So 9 months following injection, you can see in the striatum tremendous amounts of alpha-synuclein still remains. In GFP, we still get great expression using the same vector. But if you look at human alpha-syn in the striatum, you can see much greater levels in the olig-1 alpha-syn injected animals relative to GFP. Same thing we see here in terms of in the substantia nigra, and total proteins here are about the same. So in summary, for this part of the talk, intracerebral injections, olig-1 results in a robust widespread transduction. Transduction is specific to oligos. You get formation of PS, phosphoserine 129 positive resistant GCIs, potential spread of alpha-syn from oligos to neurons, and titer-dependent neurodegeneration observed in the striatum 5 months post-injection. Now, we also wanted to do similar things in non-human primates, because non-human primates are anatomically more similar to humans, and they may be better to assess translation from bench to bedside. So we basically did very similar studies in rhesus monkeys. First, we injected olig-1 GFP. This is just 1 month post-injection. You can see we injected in the caudate nucleus and in the putamen, and you can see the absolute robust number of cells that are transfected 1 month following the injection. We have almost 500,000 cells in this particular animal. And you can see the quantification number of GFP positive cells and the volume that you can get. So again, we wanted to examine the tropism that one gets in non-human primates. And you can see here that in terms of the tropism, we have tremendous expression of GFP and olig-1, but very little in NUN and very little in GFAP. And you can see quantitatively in these 3 initial monkeys that it's about 90 to 94% oligos in these animals, less than 1% or less than 2% of neurons, and less than 1% of astrocytes. So again, the tropism for these for the olig-001 is extremely strong in non-human primates. So now we injected the ALG1 alpha syn in non-human primates, and you're able to see here in the caudate nucleus and in the putamen various regions of which we have specific oligodendroglial transfection using serine 129 phosphosynuclein in the middle panel and LB509 in the right-hand panel. And you can see here in the graphing that we have over 2 million cells for the most part in all 3 monkeys that are transfected in a very large volume of our target area that is transfected. We then injected, then looked at whether these GCIs are true aggregates. So you can see an ALG2 stained cell here by the arrow. You can see LB509, you can see the merged image, and we do proteinase K digestion. And you can see most of these cells here do not digest with proteinase K. And so this indicates that these are true insoluble alpha-synuclein inclusions, just like what you see in human MSA. With regards to demyelination, you could see here that in the area of the alpha-syn injection in the corpus callosum, we see demyelination. That's not seen with GFP in the next panel. And if you put it in the striatum, you see a decrease in Luxol fast blue in the striatum, and that is also not seen with GFP. So a key feature of MSA is demyelination, and that is perfectly replicated in our animal models. There is a significant immune response in response to these injections. You can see here in alpha-synuclein, almost the entire area that is encompassed by the transduction of the vector in the lateral putamen is filled with HLA-DR stained cells, and also in the substantia nigra, where we saw the transduction spread to, we see inflammation. In terms of the GFP injections, you can see the oligo GFP regions here, and you just see basically a thin level of HLA-DR just along the needle track and not replicating what is seen in animals that received the ALG1 alpha-synuclein. So now we went on to other studies looking at 6 months post survival, and you can see here on the histology sections the areas that receive the ALG1 alpha-synuclein injections, and you can see the widespread expression of LB509, but also serine 129 phosphosynuclein is exactly the same thing. Just to show you that MSA in human MSA in the putamen, we see this phosphoserine 129, but we also see phospho-Y39, which is another factor that's involved in MSA degeneration. You can see that also in the substantia nigra, and we can also see it with ALG1 alpha-synuclein, but not alpha-synuclein with GFP. And you can see that this pathology, the PY39, is replicated in the model just as is seen in human MSA. And also in terms of the numbers of cells that we are seeing with GCIs in these studies, it's up over 30,000. At 6 months, we see this demyelination very much like we see in as I've shown you before. For brevity, I'm not going to go into that, but let's go into degeneration. At 6 months, not at 3 months, but at 6 months, we see loss of new N positive cells in the striatum, loss of TH positive cells in the substantia nigra, just like what is seen in human MSA. We have in human MSA HLA-DR, and I'm actually going to skip this for brevity. And in terms of inflammation, you can see here the quantification of human HLA-DR, striatal GFAP, striatal CD3. That's all seen in the animal model, and all of these are seen in human MSA. So to summarize, we have all these different features at 3 months that are promising, but are better at 6 months. This is all non-human primates. PK resistance, thioflavin S inclusions, phosphoserine 129, PY39, P25 alpha relocation, demyelination, neuronal loss, et cetera, et cetera, et cetera. They all mimic the human MSA. And you can, the only difference is we haven't looked for autonomic dysfunction in this model, and we haven't yet seen motor symptoms. So we're going to go with larger lesions, either for motor impairment, but we believe this model is going to be very robust and very helpful for testing out experimental therapeutics. There are biotech companies that have already come to our lab and asked us to test their compounds, and that is underway. But we're excited about using this particular model to test novel therapeutic strategies for patients with multiple system atrophy. And with that, I'll stop, and I thank everyone for their attention.